Okay. Hi everybody. Hello everyone at home. Hello everyone in the room. Thank you so much for coming out on another filthy, horrible, rainy evening. When I sit down in a minute, before I do, I'm going to close the side door for people getting chilly. Welcome to the Oxford Café Scientifique. If you haven't been before, the aim is that this should be very informal. We do know some people have fairly early buses to get. If you're in the room and you're feeling hungry or thirsty and want to get some more food or drink, please do. We are recording for people online. It also gets put out later so that people can watch it back. It'll be up to Emilia whether or not she wants it to be on our private YouTube channel, so no trolls allowed, or whether we want to make it on the public one. Either way, it's entirely up to her. The way this is going to work is we're going to have a short talk, then a short break, only 5 minutes or so in the room, and then we will start the discussion. The aim of the cafe is there is no such thing as a silly question. If you are finding it difficult to see the slides, make sure you've got the correct screen on if you're using a device. If you're in the room, you're very welcome to join the Zoom call with your audio muted, which would then mean you can look at things close up on a phone or a tablet if you wish. Okay? So basically, we're going to present everything through the online method with Zoom. What I am asking those that people stay muted, and if they have any comments or questions, if they're online, please put them into the chat. You can use that at any time. You can switch windows online any time. That's entirely up to you. The cafe's been going a very, very long time now. This is now the 24th anniversary of its very first cafe. So you're following the footsteps of Richard Dawkins, by the way. Just thought I'd chuck that in there. He taught in Blackwell's, and he told us in about, it was about, I think, 30 minutes actually. He told us how a mouse could evolve to an elephant in 1,000 generations, which then prompted the most fierce discussion ever. Since then, we've had a very, very wide range of speakers. And if you go online to the website and look down in the previous years, in the archive, you'll see a very, very large number. We've had people who've gone on to our Nobel Prizes after speaking here. Just thought I'd mention that. I've never yet managed to get anyone who's got a Nobel Prize to come and talk. I think it's the other way around. So we're clearly the breeding ground for brilliance rather than the other way. Emilia Skirmuntt is a postdoc in the Oxford vaccine group here. She also has her own company of ECS Consultation, Science Consultation. Have I got that right?
Yes.
Yes. Excellent. Maybe you should explain a bit about that if you ask her nicely. And she did academic work here before qualifying fully and is now, as I say, a postdoc. She's also very keen on this kind of public speaking about science. So I'm very, very lucky to get her to come to the cafe. Thank you very much. Over to you.
Thank you very much. I'm very happy to be here and tell you a little bit about something which I'm very, very excited about, and I was studying it also during my DPhil here in Oxford, and it's coevolution of bats and viruses. And I will set a scene first, maybe telling you a little bit about bats. Yes. So bats were seen by humans as these harbingers of death, disease, a subject of nightmares, but also there was quite a lot of bats in popular culture, in books, in comic books as well, in films. But that's, I think, a little bit unfair to bats, because these creatures are really, really remarkable, and they have many features, many characteristics, which can tell us a lot about evolution, about that's working still. Right?
Yeah.
That's okay. About biology, and also about pathogens like viruses. And I'll start with why bats are interesting. So bats are one of the oldest orders of mammals. The first known ancestor of bats, and it is the first known ancestor, so there might be older ones, is dated back to early Eocene, which is 55 to 60 millions of years ago. Apart from being the oldest orders of mammals, bats are also one of the biggest, one of the largest. There is more than 1,400 species out there of bats, and that's 20% of all classified species. The first one is rodent, by the way. They are present on all continents, apart from some isolated oceanic islands and also Arctic regions. Apart from that, they have also very long lifespan, so 10 times longer than expected given their metabolic rate. So if you imagine mouse, which has also very high metabolic rate, lives maybe 1 to 2 years. Bats, which are also quite small, microbats, so can live to 30 years. So we know that 19 mammalian species are known to live proportionately longer than humans given their body size, and 18 of these species are bats. Bats are also, they have very low rates of cancer, if any, depending on species. I will be talking a little bit about why that is, but maybe not really about cancer, but viruses, but it is connected. Apart from that, they have very important roles in the ecosystem. They are pollinators, just like bees. So, for example, without bats, we won't have tequila or mangoes. They control insect population, and they are also seed spreaders. There are two suborders of bats. One is generally capable of echolocation microbats, also known as Yangochiroptera. And these are highly specialized insectivores, but they also can eat. They can also feed on blood, they can also feed on fruit, but these are the bats, the small ones, which you can see around Europe. The other suborder are Iniptera, which is a bit of a mouthful, which are megabats or Megachiroptera. And these are also known as flying foxes, and these are a bit less specialized, herbivores, and these we see mostly in tropical regions. But something which I'm most interested in is viruses in bats. And there is more than 100 different viruses which we can find in bat species. And some of them are, you might have heard about them. So, for example, Ebola virus, coronaviruses, I guess you heard about them, and henipaviruses like Hendra virus. Bats are also most common source of rabies. Viral particles can be transmitted via saliva, so through bites, but also aerosolization of saliva, feces, and urine. Bats are also very susceptible to infection. And because of their ability to fly and migrate over longer geographical distances, which also means that they can spread the virus over larger distance, because of the roosting patterns, and because bats can tend to roost in big groups in quite contained spaces like caves and tree trunks, and also because of their ability to hibernate, which means that virus can stay undetected over the longer periods of time. Because of these persistent viral infections at a rate higher than other mammals, and also because there is evidence of asymptomatic infections, even from viruses dangerous to other animals like Ebola, like rabies, we tend to see bats as one of the major sources of zoonotic diseases. I know that you probably cannot see this graph, but I will try to describe it. So I will just tell you what zoonotic diseases are. Zoonotic diseases are infectious diseases of animals which can be transmitted to humans. And even 70% of all emerging diseases out there are zoonotic. And also on this graph, you can see that quite a few, so this is Hendra, Nipah, Ebola, SARS, MERS, quite a few of these of these viruses out there which can infect humans have their origin in bats. So why is that? Why bats are so potent reservoirs of viruses? Is bats' immune system different? Yes and no. In fact, bat immune system is not really different. There are similarities in cell populations, B cells, T cells, macrophages, immunoglobulins. All of these are the same as in other mammals. But there are differences a bit deeper than that. Because bats are the only mammals which are capable of powered flight, they are more at risk of DNA damage. Flight needs a lot of energy. It produces a lot of free radicals, oxygen species, which can damage DNA. But very similar processes and pathways are involved also in inflammation, which is present during infection. So we know, after research, we know that several DNA damage checkpoint genes, as well as innate immunity genes in bats, are either silenced or they are not even there anymore, because bats tend to dampen inflammation in their cells, just not to get their DNA damaged through either flight or infection. And again, I'm not sure how well you can see that because it's quite a complicated slide, but again, I will try to take you through it. So on your left-hand side, yes, left-hand side, you can see a human or mouse cell, which is infected with a virus. And there are several pathways, several genes involved in the response to this infection. And you can see all of it here. It's a very complicated process, obviously, because it's science. It must be complicated. But on the right-hand side, you can see a cell of one of the bat species. Again, infected with virus, there is a signal given by the receptor on the surface of the cell. But many of the pathways that you see in mouse, in human, in cat, dog, are not there, are just not present. Some of these pathways are not triggered by these danger signals. And we see more and more of these differences on this very basic genetic level there. But apart from that, I'm telling you about evolution of bats and viruses, but I want to tell you a little bit more about also something else. It will be connected to bats, don't worry, but it is something very interesting when we think about evolution of animals, even plants, any biological entity in connection to viruses. Viruses cannot be fossils. They are just genetic material packed in this protein layer. But they can leave signals in our DNA. So-called endogenous viruses or endogenous viral elements, EVs, or just integrated viruses, I might be using it interchangeably, so I'm sorry, but that's all the same things, are present in our genomes, in bat genomes, in any animal genome, in plant genomes as well. And I'll tell you a little bit about how they can get there, because that's not that easy. When a virus infects, maybe I can use that. When virus infects animal, let's say with animals, it can spread in the population, just infecting next individual and next and next. We are familiar with the process. It can cause epidemics, pandemics. It's so-called horizontal transfer. So spread via bite, via saliva, droplets, anything viral particles are in. But if this virus infects germline cells, so sperm or egg, it can get integrated into genetic material of this cell. And if this integration is not in any gene which will kill the cell instantly because it's the development is interrupted, then it can spread over generation from parent to offspring, and it can stay in genetic material of this species for millions of years. And I can tell you that 8 to 10% of our DNA is of viral origin. Most of these things are just these mutated, fragmented remnants of viral infections, ancient viral infections even. They cannot do anything. They are just these leftovers, these signals, or fossils, if you want to say that. But some of them can be co-opted and used by the host for host biological functions. So that's very similar picture, just with bats. So if viral genes got integrated, infect host and infect the germline cell, it can then spread over next generations. So we know that these integrations are in all mammals. Our hypothesis was that because bats are so potent reservoirs of viruses, these integrations there are either used more effectively to protect bats from the symptoms of viruses, for example, or there's more integrations in bat genomes. And we had a little bit of a problem looking at that when I was doing my DPhil, because the bat genomes were of very bad quality. We couldn't see anything there. So we couldn't see anything there. They were just fragments, repeats, and we knew that we need something better. And then it came BAT1K. BAT1K is international consortium, which goal is to sequence and assemble all bat genomes, all 1,400 species out there, and analyze it and see what we can find there. And this is our first paper, which analyzed, assembled 6 reference quality bat genomes. Very quickly, I will take you through that. So I'm forgetting that I have the pointer. So on the bottom, you can see our 6 bat species. On the top, you can see reference species. It's human, mouse, dog, cat, horse, cow, and pig. And you can see that apart from human and mouse, which obviously are the best quality because people work on them for ages. Well, not ages, but yes. But you can see that our bats are really, really nice and have really nice and complete genomes there. And what we did is we looked at these viruses, integrated viruses in our 6 bat species as well as reference genomes. And again, I will take you quickly through that. I hope you can see it, but I will try to describe it. So here on your right-hand side, you can see 4 different viral families, which are not very common maybe, but we can find integrations in genomes of animals from these 4 viral families. We can see that, for example, Borna virus.
It's very faint.
It is. There is Borna virus there. In humans, you can see Borna virus, but maybe not on this slide. But there's one more row here. But in other animals, you can see maybe 1 or 2. While in some of the bats, you can see all of them. These 2 bats on the bottom have all of the integrations from these 4 families. Here we looked at 3 different viral proteins, POL, GAG, and ENF. These proteins are mostly the proteins which we mostly find in animal genomes, integrations from viruses. And these colors give us different retroviral families. I won't be going into that. That's too much information. And on the top again, you can see genomes from our reference species, and on the bottom our bats. And again, size of these squares means that we found more integrations in these genomes. So, for example, GAG protein in mouse and from beta retrovirus, there is quite a lot of it. While it, for example, in I think in horse or dog, there is not that many from this orange bar. But bats are not that far off. And you can see that apart from mouse, and we know that in mouse, these integrations are all over the place, bats have more integrations than other animals. Also human is here on the bottom. And that's also remarkable if we remember that the bats have one of the smallest genomes in animal kingdom. So that's another thing which is worth remembering, seeing these numbers. Okay, but why endogenous viruses are important? I already said that these are mostly fossils, remnants, mutated, just nothing we should be worried about. Well, we don't need to be worried about them, but these genes, these viral genes, sometimes can be used by the host for some biological functions. And one of the mostly well-known of these genes is syncytin. Again, it's not a very clear picture, but what you see here is syncytia. Syncytia is pathological state which is caused by the virus, retrovirus. When virus let host cells fuse to just form this blob with nucleuses, and then virus can just easily jump from one nucleus to the other. So that is pathological state. But it turned out that this viral protein, which causes that, was used in placental mammals. So syncytin, because that's the name of this gene, is involved in placenta development. And placenta is this autonomous organ that appeared in placental mammals around 150 million years ago in one of the ancestors. Syncytin is involved in formation of the syncytiotrophoblasts by fusing cells. It is also immunosuppressive, and it is involved in muscle development. And what is most interesting, I think, about syncytin is that it wasn't just integrated into, it wasn't just co-opted from a virus and used for creating placenta in one of our ancestors, and then it was went to the another order of mammals and another. No, it was co-opted and integrated independently by each order of mammals from different viral ancestors. So this happened over and over again. Every time with different order of mammals, we find that these syncytins have different viral ancestors. What we looked at in our research, we were looking at bats, and bats have two different types of placenta, unevenly divided between megabats and microbats. Our hypothesis was that two different viral ancestors were involved in either one type of placentation or the other. So one syncytin was involved in one type of placentation, other syncytin was involved in creation of other type of placentation. At the moment, work on that is still ongoing. We found candidates in bat genomes, but we still need to do some functional analysis. Okay, so I talked about sensitins, but there are some other genes which I think you might find interesting. So some of the genes which are taken or stolen from viruses are so-called EDIs, if-derived immunity genes. These are in fact most commonly seen because this is quite easy process when host takes viral gene and just use it as immune gene to help itself to defend against exogenous viruses, so viruses which are in the environment, for example, through receptor interference, to just cover the receptors on the cell surface with these proteins which resemble the virus, but they are not viral. But there's one more, and this is ARC. Not sure if you heard about ARC. It's quite recent discovery. And discovery of this gene suggested that viruses might have been a crucial step for an appearance of self-consciousness. And it is very, very recent discovery. It was a few years back. And it uses the same strategy to jump between neurons as viruses jump between cells. And this gene is involved in processes of memorizing things, of learning. So I think that it's very interesting to think that without viruses, we might not be here. Without viruses as well, looking at sensitins, there might not be any placental mammals. Obviously, that's a bit exaggerating because nature can find a way, and sometimes if there wouldn't be a virus, maybe there would be some other gene, maybe there would be some other process. If self-consciousness, if memory, if in case of placental mammals, placenta was given advantage over some other species, then probably that might have also happened without viruses. But we need to remember that these two things we owe to viruses. But obviously that's not the whole story. Again, it's science. When studying bad immunology, we need to remember about the huge diversity of these animals. And bats are not a single entity. There is many of them, and there are huge differences in their biology, in their ecology, in these many things which will influence the connection to viruses and these processes, this coevolution which we see between them and also viruses. And studying bats can help us to find these molecular mechanisms that underlie exceptional immunity and longevity of bats. And in turn, that can help us identify these molecular patterns out there, which can be used to improve our understanding of human aging and also disease. And sorry. And also, obviously, studying bats will help us hopefully in future. I hope we learned something from COVID pandemic, also preventing future pandemics and epidemics. And thank you very much. That was me.
Thank you thank you very much indeed, Emilia. To the people online, I'm not quite sure why there's an echo. We'll have a bit of an investigate. I thought we cured our sound problems last time, but we'll have another think about it. We take 5 minutes now in the room. Please relax, and I'll turn the recording off as well. Sussex.
On that, and that would have been known before the dinosaurs disappeared. So there will have been pterosaurs in the air, and I find it strange that at that point in time, a small mammal starts to decide to take to the air and fly. So it would be great to have some context. What would have been the ecological situation in which out of the very small, out of the newly emerging mammals, some started to start flying and develop all this really sort of energetic flight with the whole things that are attached to that, with the viruses and the information suppression and the whole complications that are involved with that.
So I'm not paleontologist, I'm a virologist. I can give you time of...
molecular age as well. I mean,
so we have very, as far as I remember, it's 55 to 60 millions of years ago, and that was everything which I could find about it. Yeah.
Misinterpreted that because you said it's the oldest order of mammals.
It is oldest.
Mammals existed already.
One of the oldest orders of mammals. Yes.
Okay, that makes sense.
Okay. Before I come, I've just got to keep checking these levels get them absolutely right, because people are like, okay. Let's give that a try and see if it works. No idea.
Can I just pick up on something you mentioned very briefly, which was the low rate of cancer in bats? Can you say a bit more about that? Is it an asymmetric species with mole rats, also quite long with their size?
So I think that with mole rats, obviously there are several different animals which we know that have very low rates of cancers. Mole rats are one of them, but also for some reason squirrels. Why? I don't know. When it goes to bats, but I think that there are also different reasons for that. There are different processes, different things involved in that. It's multifactorial for sure. But when it goes to bats, a lot of these damage checkpoint genes would be also involved in cancer. So if we just get rid of inflammation genes, which can damage DNA, we also lower rate of cancer. But on the other hand, we also will let viruses to just infect our cells, and we'll be just there as a reservoir. So there are two things there. First, there are very low rates of cancer, but persistent viral infections. So that's another thing which we need to remember when it goes to that. But I think that's not the only reason for that. We are very early, like in very early stages at looking on bat biology and genomes, because we didn't have genomes before, and we are just learning now what causes it.
We have a question online going back to your kind of middle of your talk, saying somebody didn't quite get why without viruses there might not be placental mammals. I know you said it's a bit of an exaggeration, but could you recap just quickly why?
Yes, so I will go to this slide, not this slide, other slide.
For the people online, if you just look at that through the camera, just see which one.
So syncytin is this captive retroviral protein. So it is taken from a virus, from a viral infection, which got integrated into the genome of mammalian ancestor, and then it got used by this ancestor to develop placenta. And without syncytin, as we see it right now, the placenta wouldn't just fuse together, wouldn't be able to exist. because syncytin is a crucial gene, which is involved in that. And this gene is of retroviral origin.
And so that's that feature you talked about where it pulls together cells, they form like a blockage?
Yes, like a blob of cells. Yeah.
And do you think the same thing is involved in muscle?
Yes, it is also involved in this pathological state during infection with these particular retroviruses, which fuse cells together and create this like a blob of cells and nucleuses.
Thank you. Guys, because I'm on my own, I'm just going to do things in a careful order. If I ask another online question, and then we'll go back to people in the room. So a different person has this question. Could I ask, if bats are harbouring many harmful viruses for humans and animals, why are they a protected species in built-up areas?
So, yes, they do, but again, that's not every bat. And they are protected species because they are part of our natural environment. And as I mentioned before, they are pollinators. Without bats, we wouldn't have several different, I mentioned mangoes, several different plants out there. They are also controlling insect population. We would have much bigger problem with mosquitoes, for example, and malaria, if not for bats. But these are only two things. And without bats, we will have, again, there's one example which I can give. In Australia, bats, flying foxes, are reservoirs of Hendra virus. Hendra virus might not be very well known, but it is quite dangerous virus for humans, but also for horses. And in Australia, horse racing is very popular, and these horses are very expensive. So viruses from bats were infecting horses and killing these horses. Right now we have vaccine, but they were looking into killing population of flying foxes. That was a bad idea. When they started doing that, they seen that stress put on bats because they were killed in masses just resulted in viral shedding. There was more and more viral shedding and more infections. So again, we cannot forget that these are very important parts of our environment, but it's also not very smart to just decide that we will cull whole populations of wild animals in natural environment.
Thank you very much. Let me come back in the room. So normally I can stay at the side and spot what's going on. I'm trying to manage everything from my iPad today. Okay, so let me go to another question online then. So is there a model bat species which can be kept and propagated in the lab for genomic study? But the examples that the questioner puts is things like C. elegans, Arabidopsis, E. coli, etc. Aren't most of those microbes? So they're easy to propagate.
Yes. So they are. I'm aware of some labs which have just bats which they just breed and do some experiments on them, and that's in China. But I don't think that we have many of these outside. It's mostly because it's very hard to just keep big populations of animals in lab conditions, like these big animals. And we have populations of bats, quite a few in zoos, but that obviously are not lab conditions. So that's something which obviously would improve our understanding of bat biology. And it will be less dangerous for scientists who are just going into the wild and trying to sample these poor animals and getting infected with dangerous viruses. But unfortunately, it's not that easy. So at the moment, it's something which we need to do, just go and get samples from wild animals or zoos.
Yeah, and of course there are ethics about keeping animals nowadays, which are much tighter regulations for very good reason. Okay, all right, I'll come to the back.
Thank you for the talk. I would like to ask about a slide that shows with, comparing missing DNA across different species. I guess I don't understand why there's so much missing DNA in bats.
You're talking about this slide?
Yes, that one. So what exactly would cause the DNA to suddenly go missing?
That's not suddenly go. So are you talking about why bats have so small genomes, or are you talking about why they are not complete assemblies?
More why it's not complete and why it's so hard to get a clean bat genome.
So first of all, it's sampling. So it's very hard to get samples because you need to go and sample many individuals in the wild, and bats are not easy to catch, as you can imagine. Other thing was that we didn't have that good technologies when the initial sequencing and assemblies were created. And bats were also not priority. Human was, mouse was, because that was this main laboratory animal, but bats not. Right now, we are seeing that was a mistake. Bats are very important, especially when it goes to epidemiology. But yeah, that was mainly the reason. Right now, these, in fact, Bat1K right now released 100 reference assemblies of different bats. It's ongoing process, but that was our first paper, first stage of this project.
And you're spearheading.
Please,
Okay, and so you are spearheading Bat1K. It's a project that you set up.
That's not my. I didn't set it up. I was part of Bat1K, and I've done some of the analysis of this genome. The Bat1K was set up by Emma Tilling from Dublin.
From where? From where?
Dublin.
Dublin. Thank you.
No, just basically going back on viruses.
Okay.
Viruses, basically, when do they actually come into contact with the egg, the sperm? I mean, when do they actually infiltrate the mammal?
It depends. First of all, it depends on the virus. Again, like with bats, we cannot say just virus as a single entity. Different viruses have different life cycles, if we can say that they're alive, but different life cycles, different ways of infecting cells. So they can infect cells at any stage, and they can just get integrated into the DNA of the cell and just stay there. If they want to integrate into the part of the, for example, gene, which is involved in development, disrupting it, and as a result, killing the cell, then they can stay there happily for generations and just be this like fossil of the integration. But yeah, it can happen at any stage.
I mean, when it's actually splitting, you know, when it's creating the mammal in the womb, the virus would then get in prior to that.
It can. It can be at any stage. So when, yeah, it can be even before that. So when infection happens for a virus, it doesn't matter what the cell is. If there is receptor on the surface of the cell, which virus can get through, then it can happen.
And am I right in saying that viruses could get, they're so tiny they can get through virtually anything, so there's no placental barrier for a virus, the kind of immune protections we have?
Yeah, that's true. Although again, with viruses, it's hard to say all viruses can do that. There is a lot of them, and some of them are bigger, some of them are smaller. So, but in general, yes, they can get through most of the barriers if there is a receptor there.
So I wanted to ask, you were talking about the way that cells can protect themselves from such viruses by adding a protein to their coat or using it to block receptors. So it looks like the virus has already gone there, but it's not going to. Is that anything which human cells would ever exploit, or are they just not built that way?
Sorry.
So would human cells ever do that kind of thing and develop the ability to maybe coat themselves in a protein that makes it look as if the virus is already infected?
So we are not aware of any genes in human which do that. We know about genes in mouse, genes in the sheep, and there is candidate in bats, which actually is coated from filoviruses, so like Ebola virus. But we haven't found anything like that in humans. There are some other genes which I didn't mention about in humans. One of the genes which is involved in production of alpha amylase, which is in our saliva, is also coated from viruses. Also, one of the genes which is involved in inflammation and response to infection, but not as receptor interference, just response to the infection, ApoVec3 is also a gene which was co-opted from a virus and then evolved as our own gene in the end.
I guess that's evolution. If it doesn't harm us, then it carries on and perhaps evolves into something useful. Interesting. So looking around the room, just make sure that I'm capturing everybody. When you go back online, so this may be a bit further outside, but you're being treated like a virus expert now. Can you please explain how the range of viruses such as SARS, MERS, flu, COVID, RSV are resulting in the number of vaccination types we now have to have this winter? That's a very general big question.
It is a very general big question. Can you tell me again?
So the question written on the chat was, can you explain how the range of viruses, those respiratory ones I mentioned, are resulting in the number of vaccination types we're now having this winter? Is it because there's no one vaccination can deal with them all?
So that's one thing, that there is no one vaccination that can deal with them all. And there were even clinical trials looking at combining flu vaccine and coronavirus vaccine, but the effectiveness of this vaccine was very low, so the clinical trials were stopped. Unfortunately, at the moment, we're still dealing with what we got left after pandemic. The SARS-CoV-2 is still with us. Where is it moving? I'm just leaving that. So SARS-CoV-2 is still with us, and it's still dangerous. It still can mutate, and it still can change. More infections in the population means that there might be more mutations, and in the end, that can just make the vaccine useless. So preventing build-up of infections in the population protects us also from that, from these sudden jumps of evolutionary rates in a virus. I know that another thing which is introduced right now for people who are pregnant or over 65 years old is RSV vaccine. And RSV vaccine is vaccine which just got introduced in Europe, I think in the US as well. And that was ongoing trials which were going for years right now. RSV is another respiratory virus, and it was just introduced on the market, so that's why it might seem that we need more and more vaccines, but it just progress. We just get more protection over time.
I know RSV has been around for a long time, it's just now we have a vaccine.
Yes.
For the first time ever. So another rather interesting question online, I bet you this one would come up, was, can you explain, is there any evidence that bats are involved in the starting of COVID? And is there any link to pangolins? I did tell you that one would come up.
So bats are primary reservoirs of coronaviruses. And we know that coronaviruses, in general, I'm not talking right now about SARS-CoV-2, but like family of coronaviruses is in bat population. They are primary reservoir there. And so the hypothesis is that ancestral virus was present in bat population, then it mutated, it possibly jumped to intermediate species, it might have been pangolin, it might have been something else, then it jumped to humans. And we've seen this process over and over, because that's also with Hendra virus. It was bat, horse, human, Nipah virus, bat, pig, human, SARS-CoV-1, because again, there was intermediate species, MERS, it's bat, camel, human. So it's very similar process, but there is no evidence. And probably we won't see this evidence because so much time passed that it's just hard at the moment to find hard evidence out there. We can hypothesize that was how it happened because we've seen it before with other viruses, but I don't believe that we will know for sure.
And in all these cases with the respiratory viruses, the point is that they're going into an intermediate species, which somewhere in the world is close enough to humans that they can exchange viruses with the humans enough time so they can then make the leap. Is that right? Is that the way to think about it?
But we don't see most of these viruses. Viruses are happily live in their reservoir species like bats. They don't cause huge symptoms. They are just coexisting together, and that's totally normal. But because of destroying natural environment, bats are, not only bats, but all animals are moving closer and closer to humans. We have more and more contact with these viruses, and that causes these outbreaks because we also suddenly has a lot more contact with viruses which we never had contact before with. So yeah.
Thank you. Right. So we also have where are we? Why do bats have a low rate of cancer? So the questioner says he missed your explanation, and he's also incidentally asking same person, what is virus shedding?
Okay.
So two separate things, but from the same person.
I will start with viral shedding maybe because maybe I didn't explain that enough. So viral shedding is when viral particles are released to the environment from the host, for example. So through saliva, and it can be less fewer viral particles, so less shedding, more viral particles. It depends on a lot of different factors. In this case, when I was talking about that, these stress put on the animal population caused more viral shedding in this case. When it goes to cancer, I will go back because I said in the beginning, but I didn't mention that again. A lot of these processes, a lot of these pathways which are involved in inflammation can damage DNA. That damaged DNA can cause cancer. And we still don't know 100% why all, not all, but most of the bat species don't have that much cancer. We don't see that cancer, but probably that's one of the factors. Some of the pathways which are involved in inflammation are either lower or non-existent in bats.
Would it also be the case that is it possible that they are dying from other things before the cancer grows big enough to detect?
So,
I mean, do they have a lower rate of cancer than other animals?
they have lower rate of cancer than other animals. And as I said before, bats are, again, not all of them, we cannot treat them as one entity, but a lot of bats have much longer lifespan than we would expect from animals of the same size.
No wonder people are calling them vampires. That makes perfect sense now, with the mythology. Okay, I'll get back. How can you tell that a particular virus has jumped through a particular intermediate species?
Mm-hmm.
So what's the way to trace that?
So I'll show it on an example. It's not exactly an example of that, but it might help a little bit. So on the here we see phylogenetic tree, and phylogenetic trees are comparing, in this case, not, but when we are looking at ancestral sequences and trying to tell which sequence was the first one, and then which sequences stemmed from that, we are comparing mutations across genetic sequence, in this case, for example, virus, and see number of changes in these genetic sequences. We expect that ancestral sequence will have the lowest number of changes, while because of mutation rate, because of evolution, and viruses have very high rate of evolution, especially RNA viruses, we see more and more changes appearing, and we can track that. We have tools for that called molecular clocks. Accumulation of these mutations will tell us that sequence is taken, is like let's say offspring to the ancestral sequence, and we are just comparing genes of the viruses and these sequences and see, for example, from bat to pangolin to human. If we are talking about SARS-CoV-2, we can tell that they are very similar to each other. Still, what we've seen in bats is not as close to pangolin as we would expect, but they might something else happen. It might not be this exact sequence which we're looking at. It's very hard to tell at the moment.
So there could be another reservoir species out there.
It could be other reservoir species, and which, for example, infected pangolin. Or again, it's very complicated, especially at the moment, and what was happening during the pandemic.
Not made any better by everybody panicking, wanting to get answers quickly. Can you tell us, so this is obviously you're very passionate about it, and I think you're not working directly on it now, is that correct?
I don't work directly on it now. I work a little bit with collaborators on some of my, I don't know why I'm changing, on some of my work, which I was doing during the field. At the moment, I work on vaccine challenge trials and more into clinical trials and vaccines, but viruses as well.
So where do you see this work, the back and gene exchange with viruses work going? Where is it either going, where is it moving now, either elsewhere in the world, or what would you like to do if you came back to it?
Oh, I would love to continue that and analyzing the rest of bat-on-K genomes, looking at endogenous viruses, because I know that nobody else is doing that when I left. So yeah, I think that because more and more viral bat genomes are appearing out there, thanks to bat-on-K, we will see more and more patterns and more interesting things, looking at even just these co-opted viruses, and maybe we'll see some other examples of genes which are involved in biological processes of the host. So yeah.
Very interesting. Okay. I think I'm going to wind it up there just because we've got to the end of our complex questions and things, but please don't let that stop you coming to have a chat with me while I'm clearing up. My obvious thanks to Pharmacology for hosting, to everybody who's come in the room for coming out on a horrible rainy night. I'm kind of hearing no rain, so I'm really hoping it's nicer out there now. And biggest thanks, of course, as always, to our speaker, to Emilia. I found that absolutely fascinating, and I know that we went into quite a lot of kind of genetic stuff, but just the idea that genes are more malleable and exchangeable than we thought is a bit new to most of us, so it's really good to have an explanation. And thank you very much. I'm going to call you the Batwoman from now on.
Thank you very much.